Good morning, everyone. I welcome you all in my YouTube channel, that is Biology at Fingertips by Saurabh Yadav. There were lots of people that they were demanding about a simple topic which they want to study or understand this topic. They were just telling me, sir, kindly just make a short video on this topic so that you can identify that how to analyze the organic as well as the inorganic compounds that are present in case of any living tissue. So first of all, we have to identify that what is the difference between organic and inorganic molecule. So compound may be of two types. First, that will be the organic compound, and second, that will be the inorganic compound. What are organic compounds? Simply, you have to learn this, that any compound that contains carbon in them, then that compound is known as what? Organic compound. So simply, carbon containing compound are known as organic compound. For example, take the example of glucose. When you see the formula C6H12O6, so glucose contains what? Carbon. So it is an organic compound. And those elements in which carbon is not found, carbon is absent, then that element is called as inorganic element. So does not contain carbon. Okay, so that elements in which carbon is not found or simply carbon is absent, that element is known as inorganic elements. For example, sulfur dioxide, H2O, H2OS, hydrogen sulfide, any of it in which what is not found, there is no carbon is present, then they are considered as inorganic. And in which organic compound are those compound in which carbon are found. So there are the several techniques. I think these 2 points are clear. First, organic and inorganic. Carbon containing compound are known as organic, and those element or compound that does not have carbon, then they are considered as inorganic compound. Now let us proceed to the next section, that there are basically various ways through which we can identify or analyze the various type of what? Organic compounds that are present in living tissue, and the various type of inorganic elements that are found in particularly living tissue. So first of all, try to identify or analyze the technique through which we can identify the various type of organic elements that are found in case of living tissue. So our heading is how to analyze organic compound that are found in any living organism. When the question arises about organic compound, this means you have to remember in your mind that it must contain carbon containing compound. So first of all, you have to take a living tissue. You can take a plant tissue, you can take a animal tissue, you can take a microbial tissue, any tissue, but it must be a living tissue. So first of all, we have to take a living tissue. Suppose I have taken a liver cell or any vegetable. Both are, liver cell is an animal cell, vegetable is a plant cell. Any of the cell you can take that. There is no problem. You can take animal cell as well as the plant cell as you wish. Okay. Now this living cell, after taking that living cell, you have to take that living cell into a container that is known as mortar and pestle. Just like suppose there is a grinder or mixture in your home that usually help in what? Grinding so many particles, making simply chutney. Okay, that chutney, you have to make that chutney by the help of what? Basically by the help of mixer. So in case of laboratory, mixers are not used, but a simple container that is used for what? Grinding or crushing the material is basically called as mortar and pestle. So now take this living tissue, that will be liver cell or vegetable cell, into mortar and pestle. This is the structure of, this is the mortar and this is the pestle. And suppose here is the living cell or the living tissue. Okay, so with the help of mortar and pestle, we will try to grind it very properly. Before that, we have to add one extra thing, one acid that basically help in this crushing process, that is, you add one thing into this solution, that is the CCl3COOH, trichloroacetic acid. What? Trichloroacetic acid. So the second point is that now we have to take this living cell or tissue into the mortar and pestle, and then add some drops of simply CCl3COOH, this means trichloroacetic acid, and grind it very properly. When this grinding is completed, you get a paste. For example, whenever you are taking a garlic into the mixture, then after some time you get the garlic paste. In the same manner, when I have taken what? Living cell, then what I am going to get? So I am going to get the living paste. So then by the help of grinding, we get a thick slurry. Slurry basically means solid plus liquid components, or paste. Slurry, or simply we can call it as a paste of living tissue. So it may be vegetable or it may be liver cell, as you wish. Now when this slurry is obtained, as I have mentioned that it contains both some solid part as well as some liquid like particles. Now try to pass out this slurry or paste of this living tissue through the cheesecloth or the cotton cloth. So the fourth point, now this living tissue paste is passed out through the cheesecloth or simply cotton cloth, any type of cloth you can take. Then we obtain some data, and on the basis of that data we can conclude something. So first of all, note this point. This is all about the basically procedure in order to identify the organic components that are present in living organism. Now let us try to throw out this living tissue paste or through the what? This cheesecloth, or simply we can take the cotton cloth also. Now I am trying to pass out this living tissue paste from this cheesecloth. As we know that this living tissue paste contains both the particles. There are some particles whose size are really larger. So these are the larger molecules and these are the smaller molecules, and these are the pores. These are the simply pores that are present in case of the cheesecloth. So out of this is the larger molecule, this is the smaller molecule. Out of these two, which molecule will passes out through this cheesecloth? Obviously this smaller molecule will pass out through this pores of cheesecloth, and it will come from A to B side. So these are the smaller molecules. Simply these are the smaller molecules that can easily passed out from this A to B side. So simply we can say that the smaller molecules can also be called as micromolecules, and those elements whose size is larger than that of the pores of the cheesecloth that are present in the A side, they cannot passes out through this cheesecloth, then their size is larger. Their size is larger, and the elements which are passing out through this cheesecloth, their size is smaller. So on the basis of size we can call them, so those elements which are really very large, they are called as macromolecules. What we call it as macromolecules. And those molecules whose size is smaller than that of the cheesecloth, so that they can easily pass us out through this cheesecloth, that is known as micromolecule. Okay, the another name which we can give to this smaller molecule, as we know that the whole solution was made in what? A acid, that is the trichloroacetic acid. We have taken that acid. So those elements which can easily pass out through these pores and comes to the B side, they are obviously soluble in what? Soluble in acid. So the another name that is given to that micromolecules are acid soluble pool. Okay, and those molecules which cannot passes out through these pores, this means they are not soluble in case of trichloroacetic acid. So the another name that is given to the macromolecule is acid insoluble pool. Okay, now a term that is given in the NCERT, that those molecules which can be passes out through this cheesecloth and comes to the B side, that is also known as filtrate. What we call it? Filtrate. So acid soluble pool, the another name can also be called as filtrate. And those elements which cannot passes out through this pores of cheesecloth, but they are present in the A side, that are really very large, that's why they are known as the macromolecule. The another name that is given, so those elements which cannot passes out through this, called as retentate. What we call it as? Retentate. So the name which is given, there are the three different types of naming can be done. Those elements whose size is larger than that of the pores of cheesecloth, called as the macromolecules, and those elements whose size is smaller than that of the size of the or the pores of the cheesecloth, that is known as the micromolecules. Okay, and those molecules which can passes out through this cheesecloth comes to the B side, they obviously soluble in acid, because we have taken a solution of trichloroacetic acid. So it is known as micromolecule, also known as acid soluble pool. It will form the acid soluble pool. But these are the larger molecules, they are not passing out through this cheesecloth, they are considered as acid insoluble pool. So they will form the acid insoluble pool. And the another name which is given in the NCERT, that those element which passes out through this cheesecloth can also be called as, this one can also be called as filtrate. And those elements which cannot passes out through this pores of cheesecloth can also be called as retentate. Okay, note this point. Now let us see the different types of organic compound that can be found in the macromolecular section, and the different organic compound that can be found in the micromolecular section. Suppose whenever you will have to ask that kindly give me the constituent of protein, or simply we can say that protein is basically formed by combination of what, then you buddy tell sir protein is basically combination of amino acid. So amino acid, amino acid, amino acid, simply when several amino acid are repeated, then a large molecule will be formed, that is called as the protein. Simply saying that 3 amino acid, 1 amino acid plus 1 amino acid plus 1 amino acid. So when 3 amino acid combine, then it will form 1 molecule of protein. So this protein is known as the larger molecule. Since three amino acid, 1 2 3 individual unit of amino acid combining to form a larger unit, that is called as the 1 molecule of protein. Simply we can say that 3 amino acids combine to form 1 molecule of protein. This can enough. Okay, so this means what? This means these are the smaller units. Amino acid is smaller, amino acid is smaller, amino acid is smaller. So when smaller combine, then we get a larger molecule. This means the amino acid are really what? Small or smaller unit, and protein is basically a larger unit because it is formed by combination of several amino acids. So micromolecules are basically larger molecules. So here the example will be protein, and this protein is formed by combination of tiny or small amino acid. So small molecules, these are the small molecules, these are the larger molecules. So out of these two, protein is larger, so it is present in the micromolecule. Amino acids are smaller units, so they will be present into the micromolecules. Okay, or simply we can say that monomer simply means a single unit which combines to 2 3 4 5 many units combined, then it will form the polymer. So amino acid is the monomer unit, protein is the polymer unit. When several amino acid combine, then several monomer will combine. So for example, amino acid, amino acid, amino acid. This amino acid can also be known as the monomer. This amino acid also known as the monomer. This amino acid also known as the monomer. When several monomer combine, then it will form a polymer. Okay, so protein is the polymer, amino acids is the what? Monomer. Now the another example which we have to take, that when several monosaccharide combine, monosaccharide plus monosaccharide plus monosaccharide, several unit of monosaccharide combined, then it will form a polymer unit, that is called as polysaccharide. This is the larger molecule, and when three molecules are combining, monosaccharide, so monosaccharide is what? Smaller unit. So smaller unit will be present in micromolecule. So monosaccharide is present in the micromolecule, and larger molecule, that is the polysaccharide, will be present into the what? Into the macromolecule section. Now the another example which you have to discuss, that when several nucleotides, one nucleotide, two nucleotide, three nucleotide, four nucleotide, this means several nucleotide combines, then it will form a larger unit that is called as nucleic acid. Nucleic acid simply means DNA or RNA. Nucleic acid simply means DNA or RNA, deoxyribonucleic acid or ribonucleic acid. So this nucleotide, this means small nucleotide combines, then they will form a larger molecule that is called as nucleic acid. So macromolecule, in this section there will be nucleic acid, and it is formed by combination of nucleotide, then they are smaller, so here it will be nucleotide. Okay, so note this point, that example of macromolecules are protein, and example of macromolecule, that is the amino acid. Simply we can say that micromolecule example is amino acid. Several amino acid combine to form what? Protein. So protein is present in the macromolecule. The second example in the macromolecule, that is the monosaccharide. Several monosaccharide combine, then they will form the polysaccharide. So polysaccharide is present in the macromolecule. Macromolecule. Now the third example, that is the nucleotide. Several nucleotide combines to form a larger molecule that is called as the nucleic acid. So nucleotide is present in the macromolecule, and combination of nucleotide, that is the nucleic acid, present in the macromolecule. Note this point. This is about what? The elements that are found in case of acid soluble pool and acid insoluble pool. Anything. This is the acid soluble pool, this is the acid insoluble pool. We have to learn one extra example, that is lipid, but we will discuss this lipid into another section. Through this you can identify that why it is special, why it is not obeying this property. Just after some time we are going to see that. Now let us see the distribution of this biomolecule. This biomolecule can be broadly divided into two parts. The first part, that is the micromolecule, and the second part, that is the macromolecule. Okay, note this point. Now what we are dealing, that biomolecule divided into two parts. First part, that is the micromolecule, and second part, that is the macromolecule. In NCERT there is a specific distribution that has been given in order to identify that this is the micromolecule and this is the macromolecule. In NCERT a data has been given that any molecule whose molecular weight lies between 18 to 800 Dalton can be considered in as a what? Micromolecule. And any element whose molecular weight is more than 1000 Dalton can be considered as macromolecule. Then just emphasize on this point, 18 to 800, and this is more than 1000. Then the difference between 800 and 1000 is what? Near about there is no information between these 200 molecular weight. Suppose if anybody or any compound is having 950 molecular weight, that is having 950 Dalton. So it will present here or here, I don't know, according to NCERT. But whenever you will read some standard book, there is a specific distribution that has been given in some standard book for ABC or something else, that molecular weight, any compound whose molecular weight is less than 1000 Dalton can be considered as micromolecule, and more than 1000 Dalton that can be considered as the macromolecule. But note this point, because sometimes it can be asked in EAMS, because EAMS really choose the questions from the line of NCERT. So it can also ask that, it can also check that you are reading NCERT or not. So these two data for the micromolecule, but one data for the macromolecule. Okay, but remember the data that has been given in the NCERT. That's why I have mentioned both this data in order to clarify all the doubts. Okay, now as I have seen in the previous slide that those elements which are really smaller, they can easily passes out through the cheesecloth, and that is also known as what? It are soluble in acid, that is the trichloroacetic acid. So micromolecules comes under acid soluble pool. They can easily pass out through this cheesecloth. And macromolecules, those that cannot passes out through this cheesecloth, can be, or they are not soluble in acid, they are considered as macromolecule. So it is present in the acid insoluble pool. Macromolecule, where it is found? In acid insoluble pool. Now as I have mentioned that those elements or those compounds which can easily pass out through the pores of the cheesecloth can also be known as filtrate, and those larger molecules that cannot pass out, they are present into the upside of the cheesecloth because they are larger, they are called as retentate. So micromolecule are also known as filtrate, macromolecule can also be called as retentate. And now before knowing the fourth point, let us concentrate on this point. Suppose this is a cell, animal cell, outermost layer is the cell membrane, the in central portion that is the nucleus, and the space between nucleus and cell membrane is called as cytoplasm. This cytoplasm also contains two things. It contains 70 to 80 or 80 to 90%. Basically it is made up of what? Water. So 80 to 90% area of cytoplasm contains water, and this is also known as cytosol. So cytoplasm in which only water is found, just remember, cytoplasm in which only water are found, then that portion is called as the cytosol. But the cytoplasm is basically combination of both the things, cytosol plus what? Cell organelles. 80 to 90% water, but the remaining portion, this portion contains several type of cell organelles that are present in 10 to 20%. Cell organelles may be Golgi bodies, maybe mitochondria, ribosome, chloroplast, anything. So whenever, let us remember this point, whenever you are passing the components of this cytoplasm, because it is also containing some type of what? Organic compound. So when you try to pass out these components of cytoplasm through the cheesecloth, then which will pass out? Definitely water will pass out, but this portion will not pass out. Okay, so this cytosol, or the simply we can say that cytoplasm without cell organelles are present in which section? In which section? Micromolecule. Here those elements which can easily not pass out from the this what? Pores of this cheesecloth can be considered as what they are containing cell organelles. So this micromolecule, it represents cytoplasmic composition. This means it is having the cytoplasmic composition, but the point you have to note that without cell organelles. So what is present in the micromolecule region? Cytosol, that contains 80 to 90% water. It can easily pass out through this cheesecloth, so they are considered as the micromolecule. But when you are including the what, you are including cell organelles, then that cannot be passes out through the cytoplasm, so that will be considered into the macromolecule. So it represents cytoplasmic composition with cell organelles. Here all the cell organelles are present: Golgi bodies, mitochondria, ribosome, chloroplast, anything else. Okay, here all the cytoplasmic composition without cell organelles, cytoplasmic composition with cell organelles. So with cell organelles, that is the macromolecule. Without cell organelles, that are considered into the micromolecule. Now let us consider the examples that comes under the micromolecule and that comes under the macromolecule. So the monomeric unit, that is the amino acid, various amino acids combine to form what? Protein. So amino acid is the micromolecule and protein is the macromolecule. Nucleotide is the micromolecule, nucleic acid is the macromolecule. Various monosaccharide combine to form polysaccharide. So monosaccharide is the micromolecule and polysaccharide is the macromolecule. I think no question will be asked from this section, but this lipid, the question is basically asked upon this point, that is the really most important point, that lipid. Why I am telling that question will be asked upon this? Because whenever you see the molecular weight of lipid, so let us discuss something about lipid. So lipid are present in which section? Macromolecule. What I am saying? Macromolecule. Now let us discuss some point. Lipid, the molecular weight of lipid is basically in between 18 to 800 Dalton. Now the confusion arises in your mind, sir, you are telling any molecule whose molecular weight lies between this 18 to 800 Dalton, they are considered into the micromolecule, and you are writing this lipid into macromolecule. Why, sir? Why? This is not fair, sir. Why you are writing this lipid into here? Its molecular weight is 18 to 800 Dalton. So it must be present into the micromolecule. But it is not present in the micromolecule. It is a macromolecule. Why you are saying that it is a macromolecule? Why, sir? Either this is wrong, either this is wrong. So let us listen. Lipid, obviously its molecular weight is 18 to 800 Dalton. There is no doubt. But this was the cheesecloth, and basically all the things are what? Grind in the presence of acid. What type of acid? That is CCl3COOH, trichloroacetic acid. This means the solution was made of acid. So here solution is made up of acid. Okay, solution is of acid, and the retentate, or simply we can say that the paste or the microbial paste, कोई भी living paste, any living paste, contains protein, contains what? Nucleic acid contains polysaccharide. All these are not soluble in acid, that's why they are present here. Okay, in the same manner, this lipid, this solution is acid, it also contain lipid. This means lipid is also not soluble in case of acid. Note this point, this lipid is having structure like that. In class 10 you have read that there are two type of solvent. The first, that is the polar solvent, and the second, that will be the non-polar solvent. Are simply non-polar, this means OH, that is containing some carbon, containing that is known as the organic solvent as well as inorganic solvent. So organic as well as inorganic solvent that are present. Let us remember one point. This lipid contains two parts. The first part is hydrophilic head. Hydrophilic head means it is hydro. Hydro means water, philic means lover, means a water lover, and there is a hydrophobic tail. Hydrophobic, phobic means phobia. Phobia means simply you are afraid of something, so it will repel water, it will repel water, and this end is hydrophilic, so it will attract water. Philic means attraction, phobic means repulsion simply. So this lipid is basically combination of this lipid, the whole is known as lipid. So lipid is basically combination of two things. First, that is the hydrophobic tail, this means they are the water repeller, and there is a hydrophilic head, this means they are soluble in water. Or simply we can say that these lipid are not soluble, lipid are not soluble in organic solvent, for example water, for example in acid. So lipid is not soluble in acid or in water. This lipid is not soluble in acid or water, just note this point. But this lipid is soluble in inorganic solvent, for example chloroform, benzene, ether, etc., acetone, etc. Okay, this means this is the property of lipid. Lipid is not soluble in case of what? Acid or water. So because it contains a end, that is the hydrophilic lover, this means water lover, and another end, that is the water hating component. Simply you have to note this point that it is not soluble in water or in any acid, but this lipid is soluble in case of inorganic solvent. So it is not basically we can say that lipid is not soluble in organic solvent, for example water, for example acid. It is not soluble in that type of solvent, but it is soluble in case of inorganic solvent, chloroform, benzene, ether, acetone, any of it. If this solution contains any chloroform, benzene, ether, acetone, then it is soluble in that and passes out from this to this. But lipid is not soluble in acid and water. Note this point again, lipid is not soluble in acid and water. It is not soluble in acid or water, but it is soluble in chloroform, benzene, ether or acetone. Now whenever you go through this point, that this was the cheesecloth. Suppose this is a lipid. What type of solution we have taken? Just remember, what type of solution we have taken? Sir, actually we have taken CCl3COOH. This means I have taken, the solution is here is trichloroacetic acid, and this is lipid. And you have to emphasize on this point that lipid is not soluble in acid and water. In acid and water, this is the lipid, and the solution which we have taken is acid. This means lipid is not soluble in this acid. If it is soluble, then it will comes from A side to B side. But this lipid is not soluble in any type of water or in any type of acid. So it will not pass out through this cheesecloth, but it will be present into the what section? Macromolecular section. This is the micromolecular section. Again repeating, lipid is a what? Micromolecule on the basis of their weight, because its weight is 18 to 800 Dalton. So a line has been given in the NCERT that lipid is not strictly a micromolecule. This means its weight is obviously in this category, but since lipid is not soluble in any acid or in any water molecule, it is not water soluble, it is not acid soluble. This means it cannot passes out from this side to this side, because our solution containing trichloroacetic acid, and if our organic compound contain lipid, it cannot passes out through this. Okay, suppose there may be water also, so then this end can enter into this, but this is the hydrophobic tail, water hater. This will try to repel. So in presence of water also it is not soluble. Okay, so you have to remember two things, that lipid are not soluble in case of acid and water. So the solution which we have taken, that is the trichloroacetic acid, that's why lipid is not passing out from A side to B side. So lipid is present into the A side, that is known as the macromolecular section. So that's why lipid molecular weight is that. But note this point, lipid is not soluble in water and acid, so they are present into the which section? Retentate section. Which section they will form? Retentate, or simply in the section of macromolecular section. Okay, so it is present into the macromolecular section. Obviously its weight is low into the which section? Obviously micromolecular section. But since it is not soluble in water or acid, it cannot passes out through the cheesecloth, so they will be present into the where? into the retentate section, into the macromolecular section. They are not passing out. So lipid is what? On the basis of molecular weight, it is a micromolecule. But since it is not able to pass out through this solution, that is the trichloroacetic acid solution, it is present into the retentate section. So lipid are present in which section? Macromolecule section. Note this point. So again writing now, which are the points which you have to remember, that the examples of micromolecules and the examples of macromolecules. Okay, so examples of micromolecules contains amino acid, several amino acid combined to form. Here will be the protein, here nucleotide, here that will be the nucleic acid, here monosaccharide, it will form polysaccharide. But note this point, lipid comes under the macromolecule. Lipids come under the macromolecule. Its molecular weight, no doubt, its molecular weight is between 18 to 800 Dalton, but it is considered or present in the macromolecule section because it is not soluble in acid or water. It is soluble only in inorganic solvent. For example, chloroform, alcohol, etc. This means अगर हमने simply we can say that if we have taken in our solution acetone, अगर मान लीजिए acetone ले रखा होगा, then lipid may be passed. But we have not taken acetone, we have not taken alcohol, we have not taken chloroform, but our solution is what? Trichloroacetic acid. Since in the presence of acid or water, lipid cannot pass out through this cheesecloth or through this, they are not soluble into acid and water, so they are present into the retentate section, and retentate section is represented by macromolecules. Note this point, that lipid comes under macromolecule, not comes under micromolecule. So lipid is a macromolecule, lipid is a macromolecule, lipid is a macromolecule. Note this point. Now one more thing that you have to learn, because this question is very important, that lipid, the question may be asked like that, the molecular weight of lipid is 18, in between 18 to 800 Dalton, but why it is present into the macromolecular section? Then your answer will be, sir, since we have taken what? A solution of acid, and this lipid is not soluble in acid, so that's why it is not able to pass out through this cheesecloth. It is present in the retentate section. That's why lipid is considered as a macromolecule. And one another important thing that you have to remember, that some inorganic elements, what I am saying, some inorganic elements are also passed out through this through this cheesecloth when you are passing this living tissue. Okay, some amount of, basically the two most important thing which you have to remember, two inorganic elements passes out from, that is the retentate to the filtrate, because its size is very smaller. Okay, or they are, simply we can say that they are soluble in case of acid or water. Simply they are soluble in acid and water. That's why they can easily pass out from section A to B, from retentate to what? Filtrate. The two inorganic elements that comes under micromolecule, first that is sulfate and phosphate. So these are the two inorganic elements. These are not the organic elements. These are the inorganic elements. So these are the inorganic elements, that is the sulfate, phosphate. They are soluble in case of acid or water, so that it can passes out from retentate to the filtrate. So it comes under the micromolecules. These are the inorganic compounds. Note this point. Inorganic compounds. Okay. And this point, lipid. Lipid, if you see on the basis of weight, molecular weight, lipid is a micromolecule, but it is not a micromolecule, it is a macromolecule, because it is not present into the acid soluble pool, because it is not soluble in acid or water. It is only soluble in case of some inorganic solvent, for example acetone, alcohol, and some ether also. Okay, so they are present in the which section? Macromolecule section. And one extra thing that you have to remember, that sulfate and phosphate, these two are what? Inorganic elements. Note this point, inorganic elements that are basically soluble in case of acid or water, so they pass out through the cheesecloth and come to the next side. Sulfate and phosphate, these are the macromolecules, these are the inorganic compounds. Okay, so their molecular weight is also lower also. So sulfate and phosphate, these are the inorganic compounds that comes under the micromolecules. Note this point. Now let us consider the way through which we can identify the various type of inorganic elements that are present in any living tissue. So the technique that is used in order to identify the types of inorganic elements that are present in any living tissue, that technique is called as S analysis. So inorganic elements that are found in any living organism can be identified by an analysis that is known as S analysis. S simply means rock. Whenever you will completely burn anything, suppose this is a paper and you put fire, after some time what you will get? You will get the ash of this paper. So similarly, the ash analysis can be performed in order to identify this inorganic elements, the various inorganic elements that are found in case of living tissue. So note this point. Now we will see how the S analysis is done. S analysis is an analysis that is performed in order to identify the types of inorganic elements that are present in case of any living tissue. So first of all, suppose you go to any plant and after that you pluck a leaf. Then when you pluck a fresh leaf, this means it is basically heavy because it contains lots of water. Then first of all, go to the any plant and break the leaf or pluck the leaf and take that leaf. Basically the freshly plucked leaf, that is a living tissue, contains lots of water. Then weight it. Simply we can take the weight of that freshly plucked leaf. Because it is containing water in it, then the first type of weight which you have done then be considered as weight. Weight simply means weight of any compound that contains what? H2O, water. So First of all, we have taken wet weight. Wet weight simply means fresh weight of any living tissue. Suppose it is a leaf, and wet weight simply means it contains H2O. So when you pluck the leaf, frequently just weight it, you can identify its weight. Because it is containing water in it, then it is called as wet weight. Suppose whenever nowadays rainy season is going on, and suppose you are moving here and there, and ultimately rain is started, then you are cut by the rain. Similarly, your shirt will be from up to down full of water. Obviously, this means when you weight that shirt in any type of measurement unit, then that weight is basically due to that water. So the weight which is taken in the presence of water, that is called as wet weight. Okay, so wet weight, whenever you are taking wet weight, or whenever you are taking any living tissue, that is the freshly prepared living tissue, it contains both the things, organic as well as inorganic compounds. Now we are doing S analysis for only organic compounds, but wet weight containing both organic as well as inorganic. This means we have to proceed somewhere, some extra step must be taken, so that this organic will be escaped and only inorganic will be left, because our aim is to identify only inorganic components. Now whenever you come to your home, and suppose your from top to bottom your both pant as well as the shirt has been having full of water, then you put that shirt or cloth into sunlight for 1 to 2 days, then basically what happens, the water which are present in this shirt escape away. Simply means what? Escape simply means they are removed in form of vapor. So similarly, now this fresh prepared or the freshly leaf can be put into sunlight for 1 to 2 days, and in the presence of sunlight the temperature will be provided 70 to 80 degrees Celsius. Then ultimately what is going to be happen, water will remove in the form of water vapor. Maximum part will be removed. So water will remove in the form of vapor. This means all water has gone. Now the weight which you will take here, because there is no water, all water has gone in or escaped in the form of vapor, then now weight that living tissue or weight that leaf. This leaf does not contain water. This means it becomes dry. So the next type of weight which you are doing, that wet weight, after that we are putting that leaf into sunlight for 1 to 2 days. As a result of this, the water will remove in the form of vapor. So now the weight which we are taking is basically dry weight. So this is the dry weight of the leaf. But ultimately dry weight also contains organic as well as inorganic element. Again we are failed. We have to identify only organic elements, but dry weight of any living tissue also contain what? Both the things, organic as well as inorganic. Now we will take this leaf, that dried leaf, into blast furnace where the temperature is about 600 degrees Celsius. So now this dry weight or the dry leaf can be taken into the blast furnace where temperature is 600 degrees Celsius. 600 degrees Celsius in the blast furnace or the heat furnace, Basically, these are present in the industries where the large amount of temperature can be maintained for melting and casting of iron. So now take this dry weight or dry tissue of leaf into the blast furnace, and ultimately at last you will get the S. Simply means rock. Suppose this is a paper and you put fire on it. After some time, what you will get? You will get S. In the same manner, we are doing here, that we are providing 600 degrees Celsius temperature to the dry part of the leaf. As a result of this, we get S. What type of S? We have taken leaf, so it is a plant S. So this plant S only contains what? Inorganic compound. It doesn't contain any type of organic compound. All the organic compounds, for example, whenever you are providing heat 600, then carbon oxidized to the carbon dioxide and liberated into the atmosphere. Water, if some water is left, then it will be removed by the help of vapor, in the form of vapor. Sulfur is oxidized to the sulfur dioxide, and some another compounds that are present, then it will also form ammonia, nitrogen, methane will also be formed, and it also escape into the atmosphere. This means all the organic compounds, for example, that carbon, so CO2 is a carbon dioxide, basically it is a organic compound. So organic compound that are present will be escape out in the form of what? Carbon dioxide. If dry weight contains organic compound, which are the organic compound? Carbon containing organic compound. Okay, so if dry weight containing carbon, when it is provided 600 degrees Celsius, then that carbon will be oxidized to the carbon dioxide and escape into the atmosphere. Okay, this means all the organic components will be escape out. Only plant S is obtained, and in the plant S only inorganic compounds are found. So in this way we can identify the inorganic compounds that are present in case of any living cell or living tissue. So what are the different components that are inorganic components that are present in living cell? So these are the some inorganic elements, for example, sodium, potassium, calcium, magnesium, chloride, phosphate, sulfate. All these are basically given in the ionic form, and these are because it is also not containing carbon. No carbon, no carbon, no carbon, no carbon, there is no carbon. So these all are what? Inorganic elements. So plant S contains only organic compound, and out of these all, sodium, potassium, calcium, magnesium, chloride, phosphate and sulfate, the main, let us consider this point, the main inorganic elements that are found in case of plant S is calcium and magnesium. Note this point, calcium and magnesium is the main inorganic element that are found here. Now this is all about what? S analysis. First of all, we have taken the weight where water is found, and in the weight both the components are found, organic as well as the inorganic component. So what we have done? We have put this leaf, any living tissue leaf, into the sunlight for 1 or 2 days, where all the water vapor may be removed. So all the water vapor are frequently removed whenever we will put this into the sunlight for 1 or 2 days. So water is removed in the form of vapor. Then again we will take the weight, then that weight is called as the dry weight. First weight, the weight which we have done just after plucking the leaf, that is containing water, so weight. But when water is removed, then that type of weight is called as the dry weight. But it also containing organic and inorganic molecule, but our aim is to identify the only inorganic elements. Now the dry weight, or simply we can say that the dry piece of leaf will be taken into a blast furnace where the temperature may be about what, 600 degrees Celsius. So whenever temperature will be provided to any leaf 600 degrees Celsius temperature, so what is going to be happen if any organic compound, suppose carbon containing compound is known as organic compound, so any carbon containing compound, so carbon oxidize into carbon dioxide. If any some molecules of water is left here, so it can also be removed. Now sulfur, this sulfur is getting oxidized to sulfur dioxide, and several other components are also formed. H2, NH3, CH4, this means hydrogen, ammonia and methane like components can also be formed. Okay, but when this piece of leaf is totally or completely burnt into the blast furnace and all these things happen, then the part which we contain at the last, that is called as the S part, plant S part, and this plant S part contains only inorganic elements, only inorganic elements or inorganic components. Some are the example of inorganic components, for example sodium ion, potassium ion, calcium ion, magnesium ion, chloride ion, phosphate ion, sulfate ion. And out of these all these inorganic elements, the maximum amount that are found in the case of plant S is of calcium and magnesium. So I have written that when the tissue is fully burnt, this means about 600 degrees Celsius temperature is given, the carbon compound is oxidized. Carbon compound is oxidized to what? Carbon dioxide. If some water is present, so water is removed in the form of vapor, water vapors are removed, and the remaining part after this has been happened, then the remaining part are called as plant S. So the remnant called as S, that is the plant S. If you have taken a leaf, then it will be a plant S, and if you have taken any animal tissue, that can be called as animal S. Okay, so plant S contains mainly, so this plant S mainly contains which inorganic compound? These two. Out of these all inorganic elements, these two are the main inorganic elements that are present in case of the plant S. Note this point. So as in the starting section I have told you that this whole chapter, that is the biomolecule chapter, is basically concerned with the combination of two things, that is the biology as well as the chemistry. Okay, so in chemistry basically there are two type of analysis is done. The first is the elemental analysis and the second, that is the chemical analysis. Elemental analysis simply means it gives us idea about the various type of elements or the composition that are present. For example, carbon, oxygen, hydrogen, nitrogen, chlorine that are found in any living organism, then that is called as the elemental analysis. But the second type of analysis, that is called as the compound analysis, in that analysis we are trying to identify the type of organic and inorganic constituents that are found in any case of living organism. So this is all about the basic lecture about the biomolecule. If you like this lecture, then kindly like and subscribe बटन को जरूर दबाएं. Thank you all.